Welcome to the New England Journal of Medicine at the 2024 American Heart Association Scientific Sessions. I'm Stephen Morrissey, Executive Managing Editor of the journal, and I'm with Eric Rubin, Editor-in-Chief, and Jane Leopold, Deputy Editor. We're talking about a CRISPR-Cas9 gene editing with Nexiguran Ziclumeran for ATTR Cardiomyopathy study. Jane, several new therapies for ATTR, transthyretin amyloidosis, have recently been described. Where do therapies currently stand?
Well, Steve, you're exactly right. This is a pretty exciting time for new therapeutics for transthyretin amyloid. And there's been new transthyretin stabilizer drugs. There's been, transthyretin silencers, siRNAs, et cetera. And many of these are in the early phases of being tested, but nonetheless, they are all starting to uh, attack the disease using different methodologies.
This is a study using a gene editing construct, Nexiguran Ziclumeran, in transthyretin amyloidosis with cardiomyopathy. Eric, how was the study done and what did it show?
Steve, the agent, Nexiguran Ziclumeran, is a CRISPR-Cas9 construct packaged in a lipid nanoparticle. The nanoparticle fuses with target cells, releasing the genetic construct, which contains an RNA that targets the gene. It encodes an RNA that targets the gene that encodes transthyretin and the Cas9 enzyme that performs gene editing, resulting in a non-functional gene. Previous studies have shown that, as often happens with gene editing constructs, a single infusion results in marked lowering of transthyretin levels for an extended period of time. In this study, the construct was administered to 36 patients with transthyretin amyloidosis with cardiomyopathy. The primary endpoints were safety and pharmacodynamics, but the investigators also included both signs and functional tests of cardiac function. Several adverse events were reported, though many were associated with heart failure in patients who already had moderate heart failure. One patient died from ischemic heart disease. There were several measures of cardiac function that were modestly improved at month 12. Almost half of the participants showed an improvement of at least one New York Heart Association heart failure class.
Jane, as Eric says, the patients chosen for this study already had mild to moderate disease. Do you think that earlier treatment could prevent or delay heart failure?
Well, I think that that's actually possible. And so that if they treat these patients earlier on and you actually prevent deposition of transthyretin amyloid, then you can actually think that it may improve cardiac structure and thereby function. So if this can reduce transthyretin levels and prevent deposition, then uh, this should help patients delay or actually prevent the development of heart failure.
Jane, as you discussed earlier, there are now several classes, different mechanisms of action for agents that will become available for treating this disease. What more do cardiologists need to know to choose among them?
Well, I think people will need to know what is the most efficacious, which actually prevents not only heart failure, but any other adverse cardiovascular events associated with amyloid deposition in the heart. And in fact, which is the most durable over time? And for patients, what improves quality of life the most?
Thank you, Jane. Thank you, Eric. This study can now be found at nejm.org.